again, and welcome back to the e-learning course on drug-induced liver injury. In this portion of the series, there are two back-to-back podcasts on post-marketing surveillance for DILI, Lessons 3A and 3B. In Lesson 3A, we discussed large spontaneous report databases, and in this podcast, Lesson 3B, we will focus on epidemiological studies, healthcare networks, and registries as vital tools in our DILI surveillance armamentarium. And for the listener's benefit, I have no financial relationships with a commercial interest to disclose, and the views being presented here are my own and not an official position of the FDA. The information in this lesson is also provided in Chapter 5 of the SIAM's Consensus Report on the current status and future directions for drug development and the post-market setting on DILI that was issued in 2020. Greater details surrounding this topic with more background information and many supporting references can be found in the report, which is available online. As I mentioned in Lesson 3A, a key concept in post-market pharmacovigilance is that the detection and assessment of DILI risk is an iterative process that we undertake over the entire life cycle of a drug. And to this end, we use different data sources that align with different phases of this life cycle. Because of powering limitations, infrequent serious DILI events often are not observed in clinical trials. Therefore, a disciplined approach in the surveillance of post-market databases is necessary to assess a real-world association of any suspect drug with clinically serious DILI. Now, it is important to note that each pre- and post-market DILI information source has strengths and limitations. As we discussed in Lesson 3A, each of the post-marketing data sources listed here can play a complementary role in the overall DILI risk evaluation process. In this lesson, I will focus on epidemiological studies, healthcare organization networks, and DILI registries as sources of vital information on DILI risk assessment. As a broad category, epidemiological studies encompass a number of different study designs that are distinct from randomized clinical trials. Among these, case control studies compare a group of patients with DILI caused by a suspect agent matched to a group of otherwise similar patients without DILI to identify susceptibility factors or predictors of DILI. Using the case control study design, DILI cases can be obtained by referral from a large post-market treatment population and matched to age demographically similar controls without DILI. Moreover, the study design can be used to measure odds ratios of factors that are more common in the DILI group. As an example, this design has been used to identify different HLA allelic markers that predict increased DILI susceptibility associated with different specific drugs in defined demographic groups. However, because case control studies are not randomized, absolute proof of causal association of these factors with DILI is not possible without independently corroborating evidence. Case control studies are also limited in that they do not measure quantitative levels of drug exposure in the corresponding treatment population, DILI incidence, or absolute DILI risk. Now, observational cohort studies are another epidemiological study design. These are based on periods of prospective observation of all the enrolled patients exposed to a suspect drug in an inception cohort within a healthcare system. With this non-randomized study design, liver injury cases associated with a suspect drug that emerged during the observation period would typically be identified by diagnostic billing codes, and if there are linkages of utilization, that is prescription numbers, of the suspect drug across a treatment population in the system, incidence rates of these liver injury events might also be measured. Taking this a step further, in some healthcare systems, medical records of patients with liver injury cases of interest can be pulled and reviewed in order to exclude alternative causes of liver injury associated with the suspect drug, as well as to determine their clinical outcome and severity of liver injury. However, there are significant limitations with this approach. First, the diagnostic codes of liver injury for billing are often inaccurate and inconsistently applied. Moreover, documentation in the records that are pulled may be inadequate, since the records have not been specifically structured to collect DILI data. Finally, a high level of drug utilization must first accrue in cohort studies to detect rare idiosyncratic DILI cases. Now, from a statistical perspective, the measured point estimate of severe DILI incidents associated with a drug under study would typically have wide confidence intervals, since usually only a few cases would be identifiable in a large treatment population. With a need for sufficient powering to measure rare severe DILI events, some large-scale healthcare networks are being leveraged as surveillance systems. Examples of some big database platforms that are distributed across different institutions include Sentinel in the US and Midnet in Japan. These platforms contain records of large numbers of patients exposed to agents of interest in a real-world setting. As they are being developed, electronic medical records and lab data will likely become accessible for review of liver injury cases of interest. Also, future linkages with denominator prescription data for individual drugs across the corresponding treatment populations would permit measures of DILI incidence. With access to electronic medical records, it will be feasible to characterize DILI signatures associated with drugs that are hepatotoxic. On the other hand, there are important limitations to consider. Of note, the big healthcare network data are not collected for the specific purpose of pharmacovigilance, and as such, the sensitivity and specificity properties of DILI case identification might vary considerably based on the database screening and case evaluation method that would be applied. At this early phase of their rollout, any routine use of these platforms to search for DILI cases associated with any agent of concern will require further critical appraisal and validation. Finally, I want to turn to a category of post-marketing data sources for DILI evaluation that is of growing importance. It is comprised of DILI registries. DILI registries are organized systems to collect comprehensive, structured data from patients that have been referred with suspected DILI. These registries can be considered as treatment outcome registries, since liver injury associated with a drug, biological agent, or herbal product is a shared criterion for patient enrollment. Examples of DILI registries include the Drug-Induced Liver Injury Network, known as DILIN in the U.S., the Spanish DILI Registry, the Acute Liver Failure Study Group, or ALFSG, based in the U.S., the Japanese DILI Registry, the Latin DILIN, the Pro-Europe DILI Net, and the DILI-P, based in China. Based on their location, DILI registries can be reflective of different healthcare systems and, in some instances, different medicinal cultures regards to products that are commonly used by patients in their respective regions. In performing comprehensive clinical laboratory and imaging assessments of patients with drug-associated liver injury, these registries can incorporate prospective or retrospective study designs. Finally, because of the structured approaches they take in case-level data ascertainment, they can serve as an excellent research tool for DILI biomarker discovery and characterization. Nonetheless, there are important limitations to be aware of regarding these registries. Because they operate on referrals, registries typically do not ascertain all cases of DILI throughout the regions from which they collect cases. DILI registries often cannot assign a precise denominator of the utilization of the hepatotoxic drug within the referral network or region, and therefore drug-specific DILI incidence or frequency data can be difficult to ascertain. Lastly, causality methods between registries are not uniform, either in the methods that are being used or how they are applied. Therefore, it can be challenging to compare or aggregate evaluations for DILI risk across DILI registries. So let me then close this lesson by summarizing some key points that I have tried to cover. First, the reporting of cases of drug-associated liver injury with adequate clinical narratives and diagnostic information is critically important for an optimal assessment of drug-specific risk for DILI by safety evaluators and other analysts. Second, epidemiological studies, healthcare network databases, and registries, in conjunction with spontaneous report databases, all can play a vital role in the iterative DILI risk evaluation process over the life cycle of a drug. And lastly, even though each pharmacovigilance and epidemiological method has limitations regards case identification or accurate measurement of drug prescriptions in a patient population, they can strengthen DILI risk assessment and support modifications of product labeling, risk management interventions, or other regulatory actions. With that, I want to thank you for your attention and again encourage you to review Chapter 5 in the 2020 SIOMS report on DILI that covers this topic in more detail.